I'm going to continue here. We last left off on this slide right there. So we are going to talk about Mendel's experiments and Mendel's laws. So the first law that's super important is called the law of segregation. And this just says that different alleles will separate from each other when gametes are formed, and they'll end up in different gametes. So in other words, if we have this heterozygous, when this splits up into gametes, one gamete will get the capital W and the other one will get the lowercase w, and they'll segregate away from each other. Okay, that's called the law of segregation. Okay, the other one is called the law of independent assortment, and this says that different genes will assort independently of each other. And the traits for one thing won't affect the other one, and we'll definitely talk more about this one later when we start talking about meiosis. Okay? All right, so the first thing to do is talk about Punnett squares, and they're a really helpful way to try to keep track of alleles and predict, the outcome of some cross. Okay, and the one important thing to remember is that this is just a prediction, a probability based on math. Okay, and the actual outcome is random. All right, so what you do is, if we're going to cross this heterozygote with this heterozygote, each of these letters represents a gamete. So capital T is a gamete, lowercase t is a gamete. And if we go by the law of segregation, those two things are going to separate from each other. And we will write that like this. So here's the first thing. Those separate from each other. So we have this allele and this allele. And then same on this side. So we set it up as a little box. If you had bio, you're probably quite familiar with this. And then we just cross them. So if we multiply those, basically, we get this. Capital times lowercase, we get this heterozygous one. And then this capital times this one, we get this. And then this times this, we get this. Okay, I'm going to actually write this out. I think it'll be easier to see. Okay, so here's my paper. Okay, and here's my cross, T, T. Okay, and let's just say that capital T stands for tall, maybe a tall plant, and that would be the dominant allele because of the capital letter. Lowercase t is short, and lowercase by convention tells us the recessive allele. So then this one splits, and we get that T there, and that T there, and then the same thing is put over here on this side. Okay, so that's how you set up your Punnett square. Then this times this gives us that. That times that gives us that. This times this gives us this, and this times this gives us this. Okay, so now if we look at it, we can see that three of them, this one, and this one, would give me the dominant phenotype of being tall. Okay, because heterozygous would be tall, and then this one would be short. So in this case, 75% are tall and 25% are short. Okay, so that's a 3-to-1 ratio. Okay, so that's called a monohybrid cross because we're just crossing one trait. All right, so let's go back to here. Okay, so that's that. And Mendel did this to figure out what was going on with his experiments. So when he crossed this parental generation of purple and white, and he got this F1 generation, he could lay it out like this. So his true breeding purple was capital W. His true breeding white was the lowercase one, and he would get a gamete from there, which is the sperm, and the gamete from here, which is the egg. And he got them crossed to get this F1 heterozygous offspring, which was purple. He observed it to be purple. Okay? All right, and then he did this next cross of two of those purple plants. The two of the F1 generation were crossed, and he got that observation, the same mathematical ratio that we just saw of a 3-to-1 ratio. Okay, so what he's doing is figuring out what we just did. All right, so if I do this again with my purple and my white for this F1 generation, I can go back and do that again right here. Oops. Okay, if I'm going to do Mendel's cross of this one, so this is his F1 generation, okay? W, capital, lower. I'll try to make these obviously big and small. We get this. Okay, and this is how he figured out to explain that F2 generation with that 3-to-1 ratio. So these are purple, 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 and this one was white. So this was the F2 generation. This cross was the F1 generation, and they gave us, he got those offspring. Okay, so that's shown here. All right, awesome. Okay, so here's another example problem. Albinism is caused by a recessive allele, and if a homozygous man without albinism marries a woman with albinism, what genotypes and phenotypes would you expect for their children? So you should do this on a piece of scratch paper. All right, and I will also do it on a piece of scratch paper. And let's make sure we get the same thing. Okay, so I'm going to write it down while the words are up here. Albinism is recessive. So I'm just going to write this down. Okay, so a man without albinism marries a woman with albinism. What genotypes and phenotypes would you expect of their children? Okay, so the question said a man without albinism marries a woman with albinism, which makes me think of my squirrels. I have so many albino squirrels in my backyard. It's just like weird, and I wish I had some kind of paint gun or something and I could tag them and kind of keep track if they're being successful evolutionarily, and if I'm getting more of them. It sure seems like I'm getting more. But anyway, so here's our man, here's our woman, and they have children, and we do our dihybrid cross, and it turns out that every single child has 100% probability of being a carrier for albinism. So that's a heterozygous offspring who's carrying that recessive gene. Okay, so 100% of them would carry it. Then the question said if that one of their children married somebody with albinism, what is their offspring going to be? So if we take this one, this is our next cross. All right, let me just double check that I got that right. Yep, one of their children grows up and marries someone who's a carrier. Nope, they marry a carrier. Okay, what alleles would you expect for their children? So one of these kids grows up, that's this one, marries somebody that's a carrier. So now we have a heterozygous cross. What are the phenotypes and genotypes of their children? So I would get this. Okay, so now I've got a heterozygous times a heterozygous. This is going to be unaffected. These two will be carriers. This one will have albinism, so there's a 25% chance of having or being affected with albinism. There's a 50% chance of being a heterozygous carrier. Okay, and there's a 25% chance of being a homozygous dominant. All right, so in other words, 75% chance that they won't phenotypically have albinism, 25% chance that they will have albinism. Okay, and then these are their genotypes, and then their phenotypes. Okay, would be 25% with albinism, 75% without albinism. Okay? Okay, so work through that again if you need to, if I went too fast on that albinism problem. The next slide shows some of the human traits that mostly follow that simple pattern of inheritance. In other words, the dominant and recessive pattern. Okay, if we were at school, it'd be fun to look and see if we have these things. Whether your earlobes are attached or not. If your ear has a little point on it, the joints on if you have hair on the middle joint of your finger, freckles. So anyway, these are kind of fun to look at and think about. That mostly follow these simple rules of dominance and recessive. Many, many, many other human characteristics are, multi-gene, more complicated. Okay, so now we've got this little Star Wars analogy that I'm going to skip and let you look at it on your own. Mr. Sanger is a huge Star Wars fan. Me, kind of medium. Okay, and I'll pick it up on the next screencast.